Hi, my name is Jemma Sedgmond and I'm a senior lecturer at UWE Bristol. My research explores the use of non-invasive brain stimulation to improve inhibitory control, and in this video I'll provide a brief overview of my research plan titled The Role of Methodological Differences and Participant Expectations of TDCS Outcomes. So there have been a lot of studies that have used TDCS in an attempt to improve inhibitory control. However, there are an awful lot of inconsistent findings within the literature. A 2020 meta-analysis by Schroeder et al. Found a small but significant effect of TDCS on inhibitory control when looking at findings from 45 studies. However, across these 45 studies, there were many differences in stimulation parameters, including TDCS polarity, intensity of stimulation, and site of stimulation. And the research has found that the effect of TDCS on inhibitory control was moderated by some of these differences. So the first objective of this study is to compare the effects of TDCS on inhibitory control when manipulating stimulation parameters. So we will be manipulating stimulator and conduction type. Participants will receive stimulation using either a neurocon, a neurostim, or a HDC stim device. And stimulation will be delivered either using electrodes that have been coated in a conductive paste or using electrodes placed inside saline soap sponges. And research has also indicated that participant expectations can also influence TDCS intervention outcomes. So simply telling participants that they are receiving active or sham stimulation, or priming participants with statements about what the stimulation can do, can influence the outcomes of TDCS regardless of the TDCS condition administered. So the second objective of this study is to manipulate participant expectations. All participants will receive active stimulation. However, they'll be randomly assigned to one of three feedback conditions: active feedback, sham feedback, or no feedback. So all participants will receive 10 minutes of anodal stimulation delivered to the right inferior frontal gyrus, and the reference electrode will be placed above the left eyebrow. And then at the same time, participants will complete an anticipated response inhibition task. And then finally, participants will complete a standardized post-monitoring form, a survey of sensations related to the electrical stimulation, and they'll also be asked whether they thought they were receiving active or sham stimulation.